decided to start this expedition 3 years ago when I was in Mongolia in another expedition. I think that the Gobi Desert is a very isolated area of the world, and few study has been done in that part of the world. I decided that in this expedition it was very important to have a multidisciplinary approach. For this reason, I decided to involve people that were able to trap animals, that are trained to trap animals, in particular small mammals and bats. And then we need people that are able to do genetic analysis, people that are expert in ectoparasites, and people that are expert in microbiome analysis.
My lab is committed to the understanding of the role of fungi as pathogens and or potentially useful microorganisms. So we have been exploring the two sides of the fungal life. So the main question in this study that I'm posing in this study is if small mammals in this very special part of the world, pristine part of the world, have some specific fungi that are either useful or potentially harmful for men. So I see ourselves and I see the small mammals as vectors of microorganisms that are so small that they don't have legs, so they cannot move on their own. Several of these mammals are potential transporters of pathogenic bacteria and fungi.
We have the role of supporting the expedition with laboratory activity, with virological analysis performed on samples that will be taken from animals in Mongolia, in the Mongolia desert. And we are also involved in design, the protocol samplings, and help and give some information or extraction to collect sample and preserve the samples and storage them in the right way. The activity of either the diagnostic activity and research activities is performed in the area of animal and animal welfare, animal health and food safety. We are involved in the research with bats, in viruses associated with bats. Our aim is to verify the role of Italian bats as a reservoir of zoonotic viruses or emerging viruses. We found a great variety of viruses in Italian bats, including members of different genus and species like reoviruses, coronaviruses, rhabdoviruses, risaviruses, adenoviruses, and so on.
The knowledge on the bat of Gobi Desert bats are very low. At now we know about 20 species. Our main purpose is to study the really composition of the bat fauna in this expedition. Okay, to catch the bats, we will use in the night mist net like this with their pole and some different bug detectors, manual and automatic bug detectors. We have a cloth, a rope, and gear for entering the cave, vertical cave. Obviously, casque, helmet with a good light. We will find many viruses useful for our study on new viruses for Mongolian fauna and for Europe and for the pandemic in the future. In this mission, my work will be to catch some specimens of bats, for bring some sample of tissue from the wing, and the Massimo delle Donne will be the barcoding analysis for a genetical point of view.
My goal is to demonstrate that it's possible to make a paradigm shift, and instead of sending the sample to the laboratory, we can send the laboratory where the samples are. I want to join the expedition because I want to bring my laboratory in the desert. In the field, we will be sequencing coronavirus, but of course also characterizing the species that the researchers will catch there, from bacteria to fungi to insects to maybe small mammalians, and of course also the species of bats that they will collect there. Sequencing is helping taxonomy because you can identify species by looking at them, but also by looking at a special piece of DNA that we call barcode that allow classification of the living organism. We will sequence by using the Oxford Nanopore technology, that is a very small portable sequencer that allow us to sequence anywhere in the world. And this portable sequencer is amazing because the cost is so small that everybody at a certain point can buy it. And this is a DNA sequencer of yesterday. This is the DNA sequencer that we are going to bring in the Gobi Desert. This, together with the laptop, allow us to sequence whatever we want, wherever we want. There are many challenges in bringing a laboratory in the Gobi Desert. The first is the logistic. We have to bring very delicate instruments in a place where it will be very difficult not to destroy something. And also electricity is another problem. The infrastructure, we will not be sequencing in a laboratory, we'll be sequencing in a tent.
We will be in a group of 12 people, researchers, that we work in the field, Mongolian researcher and Italian researcher. This is a very important aspect of our research of our organization. The logistic aspect has been organized by these people, and without them it was impossible to organize this expedition. Also a botanist, because it's also important to have an idea of the vegetation around the oasis where we will move. We will go in 5 oases.
I'm the sole botanist taking part to the Mongolian expedition, so the reason why I was asked to take part to this is to depict, to illustrate the environment, to characterize the botanical landscape where the fauna is moving. What is exciting for me in the Gobi expedition, I would say everything, because I never visited Mongolia or the Gobi. The environment I will find there is completely different from every place I visited before, so my interests are far beyond the strict purposes of this expedition. You cannot exclude that some new species could be discovered. The main challenge for a botanist when he's working in the field is to ensure that the collected material is preserved during that short time in order to be kept home in a safe way. The typical tool we use to collect plants in the field is exactly the same, has been exactly the same since 2 centuries ago. It's a plant press. It consists of 2 rigid wood or it could be different material parts and a number of sheets of paper which has the capacity to remove the humidity from the plants. One of the reasons why you need to preserve the specimen you collect to provide material for further studies, including the molecular ones. This could be a reason why I will arrange specific method to collect, to fix, and to preserve the specimens specifically devoted for molecular analysis.
We have to sleep in tents for 2 weeks. The bathroom is the desert, and you cannot wash yourself for 2 weeks.
My feeling of living a few weeks in the desert without shower, without anything, living in a tent, I like this kind of lifestyle.
Something that for me, at least for me, it's really important is the privation of sleep. It's my sort of my Achilles heel.
The problem, but the problem are not the bats in the cave, are the bear. I hope we'll not find there.
I selected the people that will come to this expedition, and I feel the responsibility of what will happen in the field. This means that I will be relaxed only at the end of the expedition.